Hello to all, greetings of the moment. Myself Mr Karthikeyan, mentoring a NEET. Welcome to our video session. In this video module, I would like to explain about anatomy or internal structure and tissue arrangement of dicotyledonous root. So we knew already plants are classified as monocot and dicot. So monocot, root, leaf, stem, and dicot, root, leaf, stem. Anatomy topics we have in our syllabus. So I shall explain about dicot root and how the tissues organization has been taken place. So please get ready. Internal structure of dicot root. Let's see. So generally, seeds are two types, monocot, dicot. Here this is the process of germination. These are the cotyledons. Two are there, dicot. Example, sunflower. Helianthus annuus, scientific name, binomial nomenclature, sunflower. So let's see. So internal structure. This is a plant body, stem, leaves, flower. Then inside the soil, the pot is known as root. Let's see. So venation, leaf margin or lamina. Yeah, this is the root. Especially this is their students. Taproot system. So roots are two types. Fibrous root, taproot. So here we could observe mother root and also lateral roots. And also on their lateral root surface, root hair. In upcoming minutes we shall observe. So taproot, lateral roots can be observed here. Let's see. Yeah, this is known as transverse section. TS, transverse section. Taking section or cutting the object or sample transversely to the axis. So this is an axis example. Taking section transversely or horizontally is known as TS. So taking section parallel to its axis, object or sample axis is known as LS. So this is a TS. And this diagram especially ground plan. So we have two types of diagrammatic and schematic representation. One is ground plan. Second one is the entire structure, that is known as a sector. So here we could observe ground plan. This is known as ground plan. Sector nothing but if we cut in a V shape at any part or portion in the ground plan, that is known as sector. Generally in our study materials the diagrams given in the form of sector. Let's see. This one is a ground plan. Yeah, one by one we shall go through. First, this outermost layer, the cells are rectangular, closely or compactly arranged, intercellular spaces are absent. So this single layer structure is known as epidermis or epiblema. Epi means outer or upon, dermis means layer. So this first structure or outermost part of TS of dicot root is known as epiblema or epidermis. Single layer cells, rectangular, compactly or closely arranged, intercellular spaces are absent. Additionally, here and there epidermal outgrowths can be seen. So these epidermal outgrowths known as root hair. So this root hair main function absorption of water and minerals from the soil, and This epidermis main function is protecting innermost tissues, whichever they are present in the structure of the root. Let's see. Yeah, now clearly we could be. So this is epidermis, root hair, followed by cortex. So just below the epidermis, the entire tissue which is present, that is known as cortex. This central conducting part is known as stele. So the tissue which is distributed between epidermis and stellar region is known as cortex. So here cortex again 3 subparts. Exactly below the epidermis 2 to 3 layers of cells which is made by suberin. The material suberin is present in the cells, 2 to 3 layers of cells present just below the epidermis is known as exodermis or hypodermis. The main function of this exodermis or hypodermis made by suberin prevent the loss of water from the cortex to outward. Same like valves. What is the main function of valves in the human heart? Preventing the backflow, right? Same. Once water absorbed by the root hair and pass through the epidermis and enter into the cortex, from there enter into the stele, that is a pathway. But there is no chance to the water to flow back or move back because presence of hypodermis or exodermis made by suberin, 2 to 3 layers. Then next, after that exodermis, the remaining entire this green structure made by parenchymatous cells known as general cortex or parenchyma cortex. The main function is storage of food and water. Then the next one, last layer cells of the cortex, nothing but innermost layer cells of the cortex form single layer circular structure is known as endodermis. So here endodermis cells, they contains Casparian thickenings. This is a special feature of endodermis. So endodermis, general cortex, hypodermis or exodermis. These three together called cortex. Let's see. Yeah. So here root hair, expanded structure, root hair absorbing water molecules, and this entire structure is known as epidermis. Let's see how these molecules will be moving forward. Yes. So the water molecules after absorption by the root hair, they pass through the epidermis and they enter to the cortex. Then from the cortex, yeah, here we go. The water molecules are moving. So here the movement of water molecules, two types, apoplast, symplast. So symplast is nothing but slow process. Water molecules, they must cross the membranes, pass through the cells. Apoplast is a fast process. Water molecules, they never cross the cells and cell membranes. They will escape and pass through or between the cell gaps, nothing but intercellular space. Yeah, this is the endodermis. I have told you, innermost layer of the cortex form the endodermis. So these endodermis cells, they are having Casparian thickenings. The main function of endodermis is protection of innermost stellar region, nothing but pericycle, vascular bundle, xylem and phloem, and pite. Let's see. Yeah, suberin. the cast iron strips are made up of suberin. So excess of water and salts entry is prevented with the help of this endothermics. Now this is a pericycle, single layer structure, circular structure made by parenchyma cells. Pericycle main function in dicot root is it gives rise lateral roots, and also it helps in the secondary growth, especially in the dicot root, dear students. So pericycle is made up of single layer cells, parenchyma cells, and pericycle involved in the secondary growth and also lateral roots formation. But this secondary growth and lateral roots formation is absent in monocotyledonous root. That is the main difference. The remaining all points are same, structure is same. Only the thing is, in monocot root, pericycle does not involve in the secondary growth and also does not involve in the formation of lateral roots. Remember. Next here we have xylem and also phloem. So xylem and phloem together called vascular bundles. Here 4+4 arrangement is there because it is called tetrad arrangement. So 4+4. And xylem and phloem, they are arranged alternately in a different radii. In two different radii, xylem and phloem are arranged. And here we could observe protoxylem. In xylem there are two types, metaxylem and protoxylem. In upcoming topics we shall learn depth. So protoxylem towards periphery, metaxylem towards pith. Let's see. So this is a dicot root because the pith or medulla is very less or small or sometimes absent. But in a monocot, the pith is vast. In dicot, it is very small or absent. So these are the main differences between dicot and monocot root, TS of root. The thing one, dicot root, it contains pericycle. Monocot also it contains pericycle, but in dicot, pericycle involves the secondary growth and lateral root formation, but in monocot, pericycle never involve or form the secondary growth and also lateral roots. So these are the main points. And one more, in monocotyledonous root, well developed pith or medulla is present, but in a dicot, pith or medulla is absent or very less. And in a dicot root, tetrarch arrangement can be seen, vascular bundle, but in monocot root, polyarch arrangement can be seen. Apart from this, remaining all things and points are as usual. So this is a summary. Sunflower, nothing but herbaceous plant. So epiblemma, especially on the epidermis, cuticle and stomata are absent. Then cortex and stele, etc. So I hope you understood. In one more topic we shall meet. Till then, signing off, this is Mr Karthikeyan. Thank you.